All right. Hello, everybody. My name is Marco Staffney. I'm the Executive Director of the Montshire Museum of Science, and I'm here to welcome you to Health Research Live. We're going to wait a few more moments for more people to join in. We have a pretty highly registered group of folks today. We've got about 51 people on the line right now. We're excited to have you all here. If you want to go ahead and open up the chat feature on this Zoom, if you're using a desktop, that chat feature is going to be at the bottom of your screen. Just click that little chat bubble there, and it should open up a sidebar on your screen. If you're using an iPad or another type of iDevice, there might be three little dots that you can click to open that chat. And just do us a favor and let us know what town you're coming in from today. I'll put that in here. Town are you Zooming in from? Also, let us know what's your favorite exhibit at the Montshire. If you've been to the Montshire Museum before, give us a heads up. What's that favorite exhibit? Again, if you're just joining us now, we're going to get started in just a few moments. Some folks are coming on in right now. We've got a pretty well-subscribed group coming. If you're just joining us now, you want to go ahead and open up that chat feature and just let us know where you're zooming in from, and if you have a favorite exhibit at the Montshire. Bees and ants, I've seen that. So we got everybody coming out from the outside David Gotti Science Discovery Park. People coming in from all over the Upper Valley. It's nice to see folks from both sides of the river. All right, we're going to get started in just a few more moments as we wait for some more people to come back online. Bubbles, looks like Bubbles is a fun exhibit for a lot of folks here. Bubbles, of course, is one of our most popular exhibits. It's the science in soap. Soap is a very popular topic right now with all doctors everywhere. Making music is another big favorite. It's coming in right now. A lot of folks also making music on a regular basis here. Let's see, it's 2:04. I think we're going to go ahead and get started so we can keep ourselves on time. Without further ado, I think we've got a good group of folks here. And I just want to welcome everybody to this program, which is called Health Research Live. Again, my name is Marco Staffney. I'm the Executive Director of the Montshire Museum of Science and your host for today's program, Clinical Trial Research in the Upper Valley related to COVID-19. Health Research Live is a program developed in partnership with Dartmouth Synergy and Dartmouth Hitchcock Norris Cotton Cancer Center. We'd also like to thank Dartmouth Hitchcock Population Health today as well. Big shout out to our co-producers, Aurora Drew and Jenna Schiffelbein, who are both on the line too. Aurora will be helping moderate our chat section. And again, if you haven't opened that chat section, please feel free to do so at the bottom of your screen or through the little dots on your iDevice. Health Research Live is a program that looks at various sides of public health research and policy. As the COVID-19 coronavirus is the most pressing public health issue facing our communities right now, we thought we'd bring together the community around this topic. The program today will feature three different speakers, and I'm just going to take a moment to advance a slide here. All right. The program today features three speakers who will present for about 20 minutes together. Then we'll be breaking everyone into virtual Zoom rooms so that you can have smaller group discussions about what you've heard. And you'll do that small group meetings for about 15 minutes. We'll then come back together for a broader Q&A that will host the chat feature. As a note, we are recording this session, and we'll hope to be able to share this session out with CATV as well as through Montshire channels, but just giving everybody a heads up that it's being recorded. We're only recording the front presentation side, and then we'll be recording the larger and broader Q&A that will be moderated through the chat. So we hear a lot about medical trials in the news as the world is focused on finding a treatment for COVID-19. But what does this mean and what does it entail? So before I introduce our presenters, we have a poll that we'd like you to fill out to get a sense of everyone's experience with COVID-19. So it looks like our moderator here has just pulled up that poll for us. The question is, what is your personal experience with COVID-19? You know, check all that apply if you can. You either have COVID-19, you've been tested for COVID-19, someone close to me has COVID-19, someone close to me has been hospitalized, someone has died from COVID-19. I don't have any experience with COVID-19. I suspect I had COVID-19, but it's yet to be confirmed through testing. All right. 65% of the folks have participated. We'll wait just a couple more moments to see what folks have said. All right, again, filling out that poll, getting up to 83% who have voted. Looks like a majority of folks do not have experience with a direct experience with COVID-19. Few people here have had someone close have COVID-19, and looks like one person is someone who's been hospitalized for COVID-19. So it's interesting things to think about for our presenters as we move forward. And now it's my favorite time to introduce our presenters. Joining us today are two local infectious disease doctors and researchers, David DeGazel and Richard Zuckerman. They'll discuss their work. Oh, hold on one second. Getting polls thrown in my screen here. They're going to discuss their work to address the coronavirus that causes COVID-19. After they present, we'll be joined by Mary Maxfield. Mary is a retired biology teacher from Lebanon High School. Her family's experience with COVID-19 was profiled in the Valley News. She'll be speaking about her merging experience with medical trials. So David and Richard, I'll hand it over to you and let you take control of the slideshow. You can advance to the next screen.
All right. Thank you, Marcos. I appreciate it. Thank you all for coming. I appreciate the producers and the organizers for having us here. It's really a great venue. Obviously, we sort of love the Montshire Museum as someone who's lived around here for a while. So we appreciate all that you do and appreciate having this venue to present sort of what we're doing at Dartmouth and how we're interpreting sort of this current crazy pandemic that we're dealing with right now. I have control of the slides, I think. There we go. Let me put it back one more. Sorry about that. So, you know, we're just going to do very short, brief presentations here. So why are we involved in all of this research, and what do we do sort of in medical field when we're encountering disease and disability? You know, just as for all illnesses that we're involved with, we look at many different factors, and I think the SARS coronavirus 2 pandemic has really sort of strengthened our ability to really consider the major issues with research and how we approach research, because the timelines have been so incredibly compressed. You know, we really just don't have a lot of time to sort of evaluate things in the traditional fashion. So we really are spending more time thinking about how we're going about our research. And so I'll tell you a little bit about what we're doing at Dartmouth as it relates to all of these things. So I sort of think about medicine in general is thinking about it in these various settings. So epidemiology, which is the study of the distribution and determinants of disease in the human population. And these all sort of relate to each other. Having appropriate and good research and clinical information in all these venues affects the other sort of circles in this slide here. So just when the coronavirus began spreading, it's only been around since November, and in the U.S. we think since January. So it's just a very short timeline. Everyone, the need to get very good information is paramount. So we have folks at Dartmouth, so in the Department of Health in the state of New Hampshire, Elizabeth Talbot and Ben Chan, who are really working very hard on studying the risks and giving all information publicly regarding all things. Many other settings at Dartmouth are looking at this. The Dartmouth Institute, looking at sort of how various populations are infected, including the Cancer Center, looking how is the closure delay surgeries that are necessary, and what are the health effects of sort of not having as usual. So all of these things are being studied here in Dartmouth and elsewhere. looking at diagnostics, both in the clinical and laboratory setting. David's going to talk a little bit more about that. I focus my efforts mostly on looking at treatments for coronavirus. In addition, all of these have to do with prevention, because when you're dealing with treatments and you're dealing with diagnostics, they all really do sort of come into play when you're trying to evaluate and determine effective preventive methods. So, which includes staying at home, self-isolating, cleaning your hands, all the way up to and including vaccine work. So studying antibody testing, looking at which antibodies are to follow in a vaccine trial. And David will talk a little bit more about that. So I'm going to talk a little bit about sort of what I've been doing and how we go about evaluating the treatments. And I want to give you a little bit of the science about how coronavirus and the treatments work. So this is the pictorial of the SARS-CoV-2 virus. It's an RNA virus, and it has these little spike proteins on it. And these are the little S protein. S protein binds to our cells, a specific receptor that we have on our cells. And once it binds, the virus injects its material into the cell. And then our bodies, so viruses are different from bacteria, and that viruses require human cells to actually do their work. They actually use our mechanism for cellular reproduction to reproduce virus. And so that's how these viruses reproduce. So once the host cell is infected, there are potential targets that we can target for treatments. So one of the first targets you can think about is, so how do we block, how do we just kill the virus before it enters our cells? That would be one option for a treatment. How do we prevent the virus from entering the cell? As another option. There's permanent reproduction of the virus once it enters our cells. And finally, we can make reproduction inefficient. I'm going to talk a little bit about this SARS-CoV-2 and how we sort of look at that. So a couple of the things that you've heard of course, is chloroquine and hydroxychloroquine. These really make production of the virus inefficient in a test tube. And they've never really been shown specifically to work in humans. And as you know, there have been lots of reports in the literature, and this is part of the issue with research, the importance of clinical research in this setting, is really you need to get very good, adequate clinical studies done so that you can know the effects of these medications. And know I was just looking at the comments, sorry. Know the effects of these medications and if they have any detrimental effects. And as we sort of marched through this, we actually found that there were some detrimental effects once better studies were conducted with regard to these agents. The next thing that we're looking at is antibodies. So we're collecting convalescent plasma from patients who have had SARS-CoV disease, and trying to, and infusing that into patients with severe disease with the hope it binds to these proteins and makes and either kills the virus, or our body's immune system kills the virus because it has antibodies on it, or it makes entry into the cell inefficient. And finally, the last thing is that we've been looking at are the treatments that you may have heard of, remdesivir. It terminates reproduction of the virus once it enters the human cell. And this is, I'm going to focus a little bit on, sort of tell you how does a clinical research trial work when you're involved in it. So early in the course, when we figured out that clearly this disease was going to be a real problem, the research folks here at Dartmouth with and other folks approached Gilead Sciences and said, can we be involved in your trials? And we heard back from them. And once you hear back from them, you sign a confidentiality agreement, and it goes through the legal department, through the entire research organization here, of which they worked extraordinarily fast. And we had a really quick 6-day turnaround with back and forth between the company, settling contracts, making sure that we had all the appropriate regulatory components together to do this study. And then within 6 business days, we actually had this trial open and had medication on hand to enroll patients to give them the medication. Once you have that medication, really what you need to do is, once you have a COVID patient, we would sit down with either the patient or the family, depending on how sick they were, discuss the risks and benefits of the treatment, get informed consent, and if they agreed to it, we would enroll in the trial, submit their information to a central site, and then they would tell us which treatment to give the patient. Then we have it, the treatments are in a computer, in a system that sort of just opens up and we and identifies which treatment is appropriate for the patient, whether they get remdesivir 5 days or 10 days, and then we start the patient on the trial. We infuse the medication and then we follow all the toxicities and we upload the toxicities and clinical outcomes into a centralized computer, at which point, eventually, once they have enough patients involved, they evaluate that, they close the data set, and statisticians at the company evaluate the data. And that's what just happened about a week ago. The first 400 patients were evaluated in the severe COVID trial that we're involved in, and they showed that there is potentially some benefit for our patients. We still have these trials open, and that study and other studies at the NIH prompted the FDA to give emergency use access to this medication. So really, this just shows you how many components go into a strong research study, how really we need to, even in the setting of a pandemic, it's extraordinarily important to continue our ethical and make sure that our research is ethically and soundly based in clinical science. So that's just one example of all the various things that are going on here at Dartmouth. And I'm going to turn it over to David.
Thanks, Rich. Yeah, that's good. Yeah, for putting it on the next slide already. Hi, everyone. Also very happy to be part of this and love the Mount Wanshire, and glad to see that so many of you logged on. So in the next couple of minutes, I want to talk to you about another aspect of the research going on at DH. Like Rich set it up already, there is the treatment piece, and then there is the diagnostic piece, and I'm going to combine it with what you might call sort of understanding the disease, and mostly understanding how our immune system reacts to the coronavirus. So many of you might have heard that part of COVID, of the disease caused by the coronavirus, is the infection of the virus, but some people do really well and others don't. And we think that much of that has to do with how our body, our immune system reacts to the virus. And on the top graph, you can see a graph where on the y-axis, the axis going up on the side, is just sort of a generic measure of how much of a certain substance you can detect in a laboratory. And on the x-axis, you can see time pass. And there are 4 graphs in there. And the first, the black and the yellow graph, called viral RNA or viral antigen, is the number of virions, the amount of virus that you can find as people progress. Okay, so from nothing, when you just get infected, you can see it peaks over a course of a few days. And eventually, as you survive the disease, the virus goes away again. Those are the first 2 curves. Our immune system has a little lag. Initially, the virus pretty much does its own thing, unchecked. And then eventually, our immune system widens up. And one of the first things it does is it develops certain proteins called immune globulins. Okay, and they're divided into various classes. Here you can see 2, IgM and IgG. They're not specific for coronavirus. This is what happens in all viral diseases. The way that we fight a virus and ultimately become immune to a virus is through these antibodies. And one thing just to add is, and IgM is something that you see relatively early on and it goes away again, and IgG in many diseases you keep forever. I'm showing this to you because I'll come back to this in the next slide when we talk both about diagnostic testing. So you've all probably heard about serologic testing. That's what we're talking about. Antibody testing, serology, that's synonymous for this part of the immune system, okay? Now, one thing that I think sometimes gets lost, and that I want to make you aware of, and where I think the real science happens, is that, you know, each of these classes, let's just stick with the IgG, is a class. And just like a class in school, it means there are many members to the class. And that's what I wanted to show with the picture below. So below you see another cartoon of the virus with the spikes that Rich talked to you about and the RNA in the middle. And there's just 4 items that are named here, right? From the membrane protein, the spike, etc. Now, our body can make antibodies, little pieces of protein to each of these structures, okay? So therefore, you end up, once your immune system really kicks in, you end up with an array of proteins that fit onto these various proteins the virus carries. And that's where the real science happens. And that's why when you read the paper, if you read a good paper, the paper will help you understand that there's still a lot of uncertainty around the immune response, because we don't quite know yet. And Rich, if you could advance to the next slide, we don't quite yet know which of these various antibodies are really helpful. Okay, let's see if we can get, do you still have control, Rich, or maybe it sort of slowly, no, it didn't come to me yet. Okay, there we go, very good. So, right, so with that acknowledgement that there is a whole family of antibodies that we can put into classes, we at Dartmouth-Hitchcock, I'm part of a bigger group, a pretty large group actually, that extends beyond the borders of Dartmouth, that ask a couple of questions about these antibodies, okay? So some things are, for example, do we all make antibodies when we get sick? Or maybe some people don't. Why is it that young people don't get sick? Is it because they make antibodies that are so effective, or maybe they don't make any antibodies? Who knows? And when I say who knows, of course we have experience from other diseases, and in the end, this virus, while it's kicking us in the butt, it's still a virus. So there's going to be some overlap with other viruses. So we probably make antibodies, but we don't quite yet know who makes them best, old people, young people, etc. Now, because there is these various antibodies in each class, we don't quite know yet which ones are the ones that protect you. So when you go out and you get an antibody test right now and it tells you it's positive, you don't know if you actually made the antibodies that convey protection against reinfection. Okay, that's one of the things that we're still sorting out. We also don't know how long they last. In some diseases like chickenpox, you will have antibodies for the rest of your life, and they're almost always entirely protective. But in some, like the other coronavirus that give us the common cold, we know that protection only lasts a couple of years. So obviously, as Rich mentioned, it's a very new disease. We don't know yet. And then finally, and that's what I'll spend the last couple of minutes on, is how reliable are these tests? And some of it you might already have glanced at, because we haven't fully characterized all these antibodies. We just don't know how well the test works and what antibodies you're actually picking up. But another really important piece, and I love this piece because I think it really gets at what science is about. And since we're speaking in a virtual science museum and you asked us to make the link between clinical research and science, I think this is a really, really important picture. So what you see down here is a famous 2x2 table that tells you a lot about medicine. So we do a lot of testing in medicine, which you see on the left side, right? And you can either have a positive or a negative test. Well, what we're testing is the truth. We're trying to find the truth. And the truth you see on the top bar. And there is people who are really sick, right? That's the truth. They really have COVID or not. And the tricky bit is, of course, that we want a test that correctly identifies those people that have the disease or don't have the disease, okay? And there's much terminology around that. Some words that you hear thrown around is stuff like sensitivity and specificity or positive predictive values of tests. And Rich, if you would take us to the last slide, the thing that I want to leave you all with is that even if you have a really, really good test, and many of these serologic tests eventually will be really good as in they work well in the laboratory. But the one thing that I want to give you to think about is, you know, if a disease is rare, so for example, up here in the Upper Valley, we haven't had that much COVID probably. So it's not that likely that if you just pick a random person from the street that they will have had COVID. Now, if you've been really, really sick in the last month and you just couldn't get tested because there weren't enough tests, then that probability for you personally might be a little higher. But what I'm showing you here is that if a disease is rare, so if you were just to go out and test everybody with a serologic test in the Upper Valley right now, then you can see that a good test would indeed characterize many, many people as being not infected. And that's correct. They're true negatives. But on the right side, you can see that if you have one positive person in that, right, so a 1% prevalence in the population up top, that you would, with a good test, you would find that person. So that's the true positive. But you would also find two people that the test thinks are positive, but that never had COVID, okay? So in a rare disease, 1% prevalence with a good test, 98% sensitivity specificity. If you walk out of the doctor's office with a positive test, there's only a 1 in 3 chance that you actually had the disease, okay? And I think that's really, really important to realize that no matter how good the science we do at Dartmouth-Hitchcock or elsewhere, there is still just a reality that test and truth are not always the same. That's what I'm going to leave you with. All right. I want to hand this over.
Great, thank you so much, David. You're going to hand this over to me. Thank you so much. I'm just requesting the remote control right now just to advance the screen. Thank you both so much for giving us that insight, and we'll hear more from you later in our program today. Next, I want to move us to our next speaker. Mary, sorry, I'm getting back into here, making sure if I can advance the slide, which I'm having a difficult time doing. Here we go, Mary Maxfield. I'm finding you on my screen as well, because even though we do have a slide for you, I'm going to actually spotlight your video for speakers right now if they can see you, or maybe they can't. Mary is joining us today because she was featured, and her family was featured in the Valley News for their experience with COVID-19. Mary, would you mind telling us a little bit about your personal experience with COVID-19? You're going to need to unmute your microphone. There you go. Thank you. You're good.
I am good? Okay.
Yeah, yep, you're fine. I can hear you now.
Okay, great. My experience with COVID-19 is that I tested positive from a local doctor, but I'll tell you about two levels on which I experienced COVID-19. One was the course of my illness and my symptoms, and the other level was the social emotional side. I'll try to do both briefly. The course of my illness was that I got sick back on March 16th when I was traveling with some friends in the Southwest. I don't know if I actually contracted the disease here in the Upper Valley because it had arrived about a week before, that was at least that we were aware of. I was traveling with a friend from New York City, but I was also flying. And I ended up flying home to New Hampshire just a few days after I arrived in the Southwest. And I was isolated in my bedroom here in Lebanon, New Hampshire for about 2 weeks. My symptoms when I had COVID-19 were very mild. I had the dry cough. I had a relatively low-grade fever, never got over 100.5. I had the fever for about a week, never really got much above 99 even, 99.5. My primary care physician tested me after I returned to the Upper Valley 2 days after I was back. I think I was very lucky to get one of those much coveted tests just by sheer luck. And she called me about 4 to 5 days later to tell me that I tested positive. I was surprised because everybody talked about testing negative, and there I was, positive. My symptoms were basically gone about a week after I was home from my big trip. My fever was completely gone by March 26th. I felt pretty good by March 29th. And I was told by my doctor and also by the Department of Public Health, New Hampshire Department of Public Health, that I was golden, that I could leave my bedroom, that I could that I could do things like the regular social distancing and precautions that everybody else was beginning to do. I could go to the supermarket for those brief little visits, take a walk outside, etc. I was really happy, but that's when the social emotional stuff comes in. I was hoping that after I was declared all better, all recovered, that I'd be freed up so that I could be of service to other people. I really wanted to work at Meals on Wheels, for example. And I also wanted to go work at the Haven, which I had worked at a little bit. But by that point, it was becoming clear as I was reading just sort of popular science articles in the New York Times, et cetera, that there was a lot of uncertainty about everything, about the degree to which was I still shedding the virus, for example? Would I be a totally safe visitor to the people who would need Meals on Wheels, a very vulnerable population? Was I really immune? Did I have active antibodies? So I didn't know. I knew that a lot of people who did test positive for COVID were asymptomatic. So I suddenly was in the disappointing position of feeling as if I needed to basically stay quietly at home the way that everybody else was without going out and doing service. So that's my story. That's my experience with COVID-19.
And Mary, maybe you could tell us a little bit about your experience now with trials on COVID-19.
Well, yes, and beginning tomorrow, I'm going to begin day one as a participant in some trials at Dartmouth-Hitchcock, 2 different studies that are going on there. One is a study that is being run that's entitled Immune Responses to the Coronavirus, that's going to be exploring a lot of the questions that David and Richard were discussing about antibodies, the part of the virus that my body might be making the best response to. Whether or not I even have active antibodies that make a difference, that's not sure, for anybody, but certainly for a 65-year-old, et cetera. I'm also going to be in David de Geysel's study, and I'm sort of excited about that one because again, my antibodies are going to be tested quite a bit. And I'm also going to be donating some DNA, I believe. And David, correct me if I'm wrong. I'll be donating some DNA to evaluate whether I have certain genes that might be associated with a mild or a more severe response to the coronavirus. So that's really pretty cool. The reason why I wanted to do that was partly for the sort of the altruistic, serving the community sense that I think a good clinical trial does, serving the world sense. Also, I was a high school biology teacher for my entire adult life. I taught students about scientific process, had my students use scientific process. I love the idea of being in studies. I like the idea of being a data point. And also there's a certain self-serving piece here, which is that I would love to indulge a little bit in asking people some questions as I'm sitting and giving. I'll be donating plasma, not in those studies, but in just a regular plasma donation thing that a lot of people with COVID are doing at DHMC and hospitals all over the world. But I'm hoping to answer some of my own questions. What about this? What about that? Can I do this? So that's my story.
Awesome, Mary. Thanks for being so willing to do that, and for telling your story, and for being so continuously curious. And thank you for all your years of service as a biology teacher. We love our science teachers here in the Upper Valley, so we really appreciate it.
You've inspired me.
Great. So now, as part of our program, we're going to break up into small groups for about 15 minutes. If you've never gone into a small group in Zoom, what will happen is your screen will disappear. It will go boop, 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 and then you'll pop into another set of screens with like only 4 or 5 people. If you're not using your video, this is a good opportunity to unmute yourself and put your video on so that you can see those other people in your group to have a small discussion. When you get in there, you might want to quickly introduce yourself and then just tell people about what brought you here to this event today. You're going to also want to answer, in your experience, how is COVID-19 research impacting your community, if it's having any impact at all, and what questions do you have?
And looking at antibody responses is going to help determine, what those antibody responses are that are important to look for in patients who are vaccinated, because testing a vaccine is also difficult in this time where you might have to give someone a vaccine and you're looking at the immune response, but you really don't know if that immune response is protecting them from getting the virus. You just have to sort of see if they acquire the virus, if they're in the setting where they would normally do it. And that requires a lot of patience and a lot of time. And with this pandemic, obviously, these times are trying to be compressed, but that's the difficulty here and why the timelines are so long for a vaccine to look at efficacy. And with regard to treatment trials, we have been doing, I saw the question about placebo, and remdesivir has been tested against placebo as well.
I'll also add one more thing. You know, when you think about treating somebody who is sick versus protecting people who are healthy, the safety standards also goes up quite a bit, right? So if you're really sick, then maybe you're willing to take a chance on a treatment. But if you're otherwise totally healthy, you shouldn't be asked to take a chance on incurring harm from a vaccine that's meant to protect you.
Right. I'm trying to sort of sort through all of the questions and figure what is a next logical question. I think one of the things that that came out as pretty explicit is exactly how does remdesivir work? Like what are we looking for it to do?
Yeah. So remdesivir works by entering the cell, and it actually incorporates itself. It looks like one of the building blocks of RNA and DNA. And so what it does is it wiggles way into the process that the virus is using of reproducing that strand of RNA so that it can produce more RNA and make new virus. And so what remdesivir does is it poisons that process and terminates it. So that the RNA can't reproduce, and so new virus can't be produced in our body. Now it's excellent in a test tube, but sometimes in real life, you know, as we know, viruses are a little bit squirrely, and they can figure out mechanisms to get around that. And so that's why you need absolutely to test in human trials to make sure that it does what you think it's going to do. And so in order to test that, what we have to do is, you know, number one, make sure it's safe. Number two, determine if there's efficacy. And the comparative trials that we've been looking at are mostly in severe disease, but we also have a moderate disease trial, patients who aren't as sick. The reports that have come out have been in the severe disease trial. The NIH trial was a placebo-controlled trial that showed some efficacy at improving patients clinically so they're discharged from the hospital sooner. And there may be a benefit in less deaths, but we're not sure. These studies haven't even been published yet. This is just new data that Dr. Fauci and Gilead has presented. So we really are waiting for these truly peer-reviewed publications to determine what, if this actually does work. It looks like it may, but we're not 100% sure at this point in time. So in that setting, we're also looking at safe in addition to either improving disease faster or having less people die or have very severe illness.
So one question that's been in the news a lot I'm seeing is about mutations of COVID-19 and will it mutate, is it mutating, has it mutated already? Could you just do a little explanation about what that actually means, and then how does that throw a wrench or benefit the type of clinical research that you're already doing?
I'll let David talk about that.
Yeah, sure. So we all living beings mutate, and virus, and especially an RNA virus like coronavirus, mutates even more, okay? More easily, I should say. And there's a whole biological reason for that. But I think what's really, really important to realize is that most of these mutations are not clinically significant. So think about your family. You all share a lot of genetic material, right? A lot of DNA is very similar in all of you. There are some differences there. You could call them mutations, and they don't make you different people, essentially. And that's what we're seeing with the coronavirus. If you pick the virus up from different people and you sequence them, meaning you figure out each letter in the RNA of that particular virus, you will find certain letters to be different, and those are called mutations. But so far, we haven't found that any of these mutations are in parts of the viral RNA that make the virus behave differently. So there's no evidence that there are strains that are more aggressive or that have become resistant to remdesivir. And therefore, clinically, we don't think any of these mutations are relevant right now. Okay, we use them, or other researchers use them, to build really nice networks of viral spread, so you can figure out who infected whom. You might have seen the New York Times today has a cover story about how most of the seedings in the United States, the virus has been seeded in the United States, mostly from New York City. That's based on mutations in the virus. But once again, clinically not that relevant. Now, does that ultimately affect our immunity, and would it affect a vaccine? We don't know yet. It could be, in theory, right? So if one of these mutations, for example, happens in the part of the virus that codes for the proteins, the spike protein or others, then in theory that would be possible, but we don't know yet.
Thank you. One more thing from the news that coming into me privately to help with any kind of clarification. I think a lot of people on this call work with children. And so there's a lot of talk right now about coronavirus and Kawasaki syndrome that's coming up in the news. I know that how would one even start to approach medical trials for that, or figuring out whether their things are correlated, or how one would treat that?
Yeah, this is an incredible example of where research is super important, right? You have some physicians who are noticing like COVID toes or this new illness, Kawasaki-like illness, and now there are all these sort of registries and various other, email threads that are just abuzz with various cases of things. And once people start to put that together, that's when the research mechanisms need to begin. So we need to accumulate these cases. We need to determine, what is it about these people that's different? Some of these, some of the patients with, the COVID toes they're talking about don't even have positive tests. Why would you maybe get that without a positive blood test? With the Kawasaki's, that's an extraordinarily new process. We think that there's probably a significant amount of disease that occurs in children asymptomatically. And there probably is some evidence in certain settings that children probably transmit virus throughout communities as well, which is one of the important things as we start to open things up, to really consider what contribution do various populations have to spreading more of the virus once we start to open up. But now with the Kawasaki-like syndrome, the various centers, New York City, the Public Health Service and Britain are putting together the cases. And then as epidemiologists, we do extensive histories to determine what sorts of exposures, what sorts of other medical histories. And then that's when you also can get samples and look at genetics. And even, is there a genetic predisposition to this, or is there some other process? And so well-identified research is going to really point to maybe who gets this. But at this point, it's so new. And the cases are just, 60 so cases now, from what I've heard, have been reported.
Sorry, we have just like a few more minutes that people give us. I'd like to sort of finish on one sort of overarching question that I'm getting, and I'll let Mary actually start from a patient's perspective, and then I'll let the two doctors chime in afterwards. Restrictions are being released everywhere right now, and Governor Scott, of course, has been releasing at a rapid pace yesterday, and more expected tomorrow. New Hampshire's had all sorts of different releases of restrictions. Mary, from someone who's had it, like, I'm just curious what your thoughts are about the releasing of some of these restrictions, and then I'll ask the two doctors on that one. Mary, you're muted. I'm going to unmute you. You are unmuted now. Go for it.
I tend to be fairly not risk averse, and with the onset of COVID-19 and seeing what it's doing to everybody in our country and in this area, I've become very cautious. So that it seems to me that even though restrictions are being lifted, there's still really strict rules about 6-foot distancing, being in areas like in groups of 10 or less. And so I'm not sure how radically different things are. I mean, all of us can still have been able to go outside to take walks by ourselves. So I guess that I have mostly questions like, gosh, what's new? Probably for workplaces, for commercial, this is very critical. For social and family, I'm not sure how different it is, or at least I don't think it doesn't feel that different. So that's my reaction.
Thank you. Richard, Davi?
Yeah, I can, a lot of this I think is very opinion based, and obviously there's a lot at stake, right? First of all, I think we shouldn't underestimate the price that many of us have paid economically, socially, emotionally with the lockdown. Really important to me is that this idea that the lockdown was really meant to buy us time, right? Never, or I hope nobody really thought that we were going to be locked down until we have a vaccine or until the coronavirus goes away, because it would be devastating to the world. So what did we need time for? I think we really needed time to have a very solid test and trace network where we can test anybody who was even slightly ill or has been exposed, and then trace people that they've been in contact with. Do we have that? It's not perfect yet. I do think Vermont and New Hampshire have come a long way in that, much further than other states. Is it sufficient? I think time will tell. I don't know, Rich, if you have a better opinion on that, but I don't know. I think we'll have to try.
Yeah, I mean, I think I agree with you completely, David, as far as getting systems in place and making sure the medical system's not overwhelmed, and also making sure that, even if restrictions are lifted, that we really practice appropriate hygiene, that we make sure that, we all understand and know how this virus is transmitted and how we can prevent it from being transmitted. And just being extraordinarily careful. It's, I don't think we should just sort of all of a sudden lift restrictions and say, all right, we're done. Life is going to go on similarly. It's going to be a slow integration. I really think it's important to realize that.
Thank you. Thank you so much for all of your thoughts on this is a very pressing topic for everybody, I'm sure, on this call. A lot of questions about very specific items, and I just wanted to point people to a resource that I've been participating in, which is Dartmouth Hitchcock's ECHO sessions, ECHO like how you would spell echo, that you can look up if you just search on DHMC's website. Every week they've been doing programs about COVID-19 as it relates to workplace and environments, and that's where a lot of these shifts are starting to manifest as people are going back to work, and it's the science of how we get back to it. So a lot of information about cleaning surfaces, a lot of information about masks and things are going to be coming up in weeks ahead. So I would just point folks to our local hospital in thinking about what kind of information might be put out there. They've been very useful and helpful sessions as someone who's thinking about how to restart. In the chat, I did put up a survey just to let us know, as we like to do more of these Health Research Live programs. I just want to thank you personally, David, Richard, and Mary. Thanks for everyone who attended today's session. I think we've learned a lot and, as of course, come out with more questions. Everyone who did pre-register for this program will receive a follow-up email with a link to the survey that I put in the chat. But if you want to take it right now, feel free to click that button and that will take you to a survey. Just a very brief 5-minute survey. I appreciate everybody's time and attention to this. Thank you so much to all the producing team and all of the help that we've had, and to our speakers. On behalf of the Monshire, I just appreciate everybody being here, and we hope to see you soon here at the museum. Not too soon, but soon.